Welcome back. In addition of the previous videos, we were talking about bacterial toxins. A toxin or poison is a specific substance, often a metabolic product of the organism that damages the host. Toxins can even induce diseases in the absence of the organism that produced them. Diseases can result from the entrance of a specific toxin into the body of a host. This is called intoxication. The term toxemia refers to the condition caused by toxins that have entered the blood of the host. Toxins produced by organisms can be divided into two main categories: exotoxins and endotoxins. Source of exotoxin: certain species of some gram-positive and gram-negative bacteria. And endotoxin: cell wall of most gram-negative bacteria. Secreted from cell: yes. Endotoxin: no. Chemical nature: polypeptide. But endotoxin: lipopolysaccharide. Location of genes: plasmid or bacteriophage in case of exotoxin. Endotoxin: bacterial chromosome. Then toxicity: high. Fatal dose on the order of 1 mg. Low fatal dose on the order of hundreds of micrograms. Then antigenicity: induces high titer antibodies called antitoxins. And poorly antigenic: vaccines, toxoids used as vaccines. No toxoids formed and no vaccine available. Heat stability: destroyed rapidly at 60 degrees Celsius except staphylococcal enterotoxin. Stable at 100 degrees Celsius for 1 hour. Important bacterial exotoxin and their mode of action. So different diseases are given: diphtheria, tetanus, botulism, gas, gangrene, anthrax, toxic shock, scarlet fever. These are gram-positive diseases. Again, gram-negative: diarrhea, bloody diarrhea, cholera, whooping cough, Escherichia coli, Vibrio cholerae, and Bordetella pertussis. So next is about control of microbial growth. Control of microbial growth means to inhibit or prevent growth of microorganisms. This control is effected in two basic ways: by killing microorganisms or by inhibiting the growth of microorganisms. Control of growth usually involves the use of physical and chemical agents which either kill or prevent the growth of microorganisms. Agents which kill cells are called sedal agents, and agents which inhibit the growth of cells are referred to as static agents. Thus, the term bactericidal refers to killing bacteria and bacteriostatic refers to inhibiting the growth of bacterial cells. Terms commonly used to describe antimicrobial agents and processes: antiseptics, a disinfectant that is non-toxic enough to be used on skin. Examples include alcohols like ethanol, 50 to 70 denatures proteins and solubilizes lipids. Silver nitrate precipitates proteins. Iodine solution inactivates protein. Silver nitrate can be used like that. Oh, that's a brand new knowledge for me. Examples include alcohols: ethanol, 50 to 70% denatures proteins and solubilizes lipids. Silver nitrate precipitates proteins. An iodine solution inactivates proteins. Detergents disrupt cell membranes. Then disinfectants, a chemical used to destroy many microorganisms and viruses. Examples include hypochlorites, chlorine compounds, copper sulfate, formaldehyde. Wait a minute, I'm gonna read the antiseptic one once again. A disinfectant that is non-toxic enough to be used on skin. Examples include alcohols, ethanol denatures proteins and solubilizes lipids. Silver nitrate precipitates proteins. Iodine solution inactivates proteins. Detergents disrupt cell membrane. And disinfectant, a chemical used to destroy many microorganisms and viruses. Examples include hypochlorites, chlorine compounds, copper sulfate, Formaldehyde reacts with NH2, SH, and COH groups. Phenolic compounds denature proteins and disrupt cell membranes. And mercury chloride inactivates protein by reacting with sulfide groups. Sterilization. Sterilization refers to the complete and total destruction or elimination of all viable organisms, including microbial endospore in or on a substance being sterilized. Sterilization procedures involve the use of heat, radiation, or chemicals, or physical removal of cells. Pasteurization. Pasteurization is a brief heat treatment used to reduce the number of spoilage organisms and to kill disease-causing microbes. It uses precisely controlled heat to reduce the microbial populations in milk and other heat-sensitive liquids. The process is named after Louis Pasteur, who first used heat for controlling the spoilage of wine. Pasteurization does not kill all organisms and is therefore not synonymous with sterilization. Milk is usually pasteurized by heating, typically at 63 degrees Celsius for 30 minutes. Batch method, or the 71 degrees Celsius for 15 seconds. Flash method to kill bacteria and extend the milk's usable life. Microbial agents. Physical agents. Heat, radiation, and filtration are most common physical agents normally used to control microbial growth. Heat is the most rapid and effective physical agent used to control a population of microbes. Both dry and moist heat is used to kill microbes. Moist heat generally coagulates and causes denaturation in microorganisms. Heat, radiation, and filtration are most common physical agents normally used to control microbial growth. It is the most rapid and efficient physical agent used to control a population of microbes. Both dry and moist heat is used to kill microbes. Moist heat generally coagulates and causes denaturation in microorganisms. In dry heat, by contrast, the primary effect on microbes is due to oxidation of large molecules. Similarly, there are many different kinds of radiations having antimicrobial effects. Ionizing radiations, which include X-rays and gamma rays from free radicals in cytoplasm, and the free radical damage microbial protein and DNA. Other important form of radiation is ultraviolet radiation. These radiation damage nucleic acids. Filtration involves the physical removal of all cells in a liquid or gas. It is especially important for sterilization of solutions which would be denatured by heat. Antibiotics, amino acids, vitamins, etc. In this process, solutions or gases are passed through a filter of sufficient pore diameter, generally 0.22 micron, to remove the smallest non-bacterial cells. Then chemical agent. Large number of chemicals such as alcohol, aldehyde, heavy metals, halogens, and dyes are commonly used as antimicrobial agents. Alcohols like ethyl alcohol and isopropyl alcohol causes denaturation of proteins and used as an antiseptic. Two heavy metals, mercury and silver, are used as antimicrobial agents. Mercury is used as mercury chloride and as a component of mercurochrome. Silver is used as silver nitrate. Two important halogens, chlorine and iodine, act as oxidizing agents that react with amino acids and proteins and change the nature of the protein. Similarly, gases like ethylene oxide, formaldehyde, and beta propionolactone are used to achieve sterilization. Then antibiotics. Antibiotics are low molecular weight antimicrobial agents produced as secondary metabolites by microorganisms that kill or inhibit other microorganisms. The term coined by Selman Waxman originally described only those formulations derived from living organisms. A more broadened definition of an antibiotic includes any chemical or natural origin which can kill or inhibit the growth of other types cells. Since most clinically useful antibiotics are produced by microorganisms and are used to kill or inhibit infectious bacteria, we can follow the classic definition. Antibiotics may have a sedal effect or static effect on a range of microbes. The range of bacteria or other microorganisms that are affected by a certain antibiotic is expressed as a spectrum of action. Antibiotics effective against a wide range of microorganisms such as both gram-positive and gram-negative bacteria are said to be broad-spectrum antibiotics. If effective mainly against gram-positive or gram-negative bacteria but not both, they are narrow spectrum. If effective against a single organism or disease, they are referred to as limited spectrum. Antibiotic molecule produced by a microbe that are subsequently modified by an organic chemist to enhance their antimicrobial properties or to render them unique for pharmaceutical patent are described as semi-synthetic antibiotic. Antibiotic molecules produced by a microbe that are subsequently modified by an organic chemist to enhance their antimicrobial properties or to render them unique for pharmaceutical patent is described as semi-synthetic antibiotics. Antibiotics inhibit microbial populations by any of five major methods. They disrupt cell wall synthesis, they interfere with cell membrane function, they inhibit protein synthesis or interfere with its completion, they disrupt the functioning of nucleic acids, and they interrupt selecting metabolic pathways selected. Microbial sources of some antibiotics. Sulfonamides or sulfur drugs. Sulfonamides or sulfur drugs are structurally related to sulfanilamide. Sulfanilamide, an analog of p-aminobenzoic acid. The latter substance is used in the synthesis of the cofactor folic acid. When sulfanilamide or another sulfamide enters a bacterial cell, it competes with p-aminobenzoic acid for the active site. It results in decrease in folate concentration. The decline in folic acid is detrimental to the bacterium because folic acid is essential for the synthesis of purines and pyrimidines. Penicillin was first discovered by Alexander Fleming as a metabolic byproduct of the fungus Penicillium octatum. All penicillins share a common bicyclic nucleus that is composed of used beta-lactam and thiazolidine rings. The most crucial feature of a molecule is the beta-lactam ring, which appears to be essential for activity. The mechanism of action of penicillins is still not completely known. It has been proposed that penicillins inhibit the enzyme catalyzing the transpeptidation reaction because of their structural similarity, which would block the synthesis of a complete fully cross-linked peptidoglycan and lead to osmotic lysis.